In the year 2010, Deepwater Horizon, an offshore drilling rig, experienced a blowout leading to the release of 4.9 billion barrels of crude oil into the Gulf of Mexico. As a result of the spill, a deepwater hydrocarbon-enriched plume was formed during the multi-phase injection of gas and oil from the wellhead. A plume formed somewhere between 1,000 meters and 1,300 meters below the sea surface and consisted of light alkanes. It was categorized by local oxygen depletion, elevated concentrations of low molecular weight polycyclic aromatic hydrocarbons, and methane. In late May 2010, the plume-associated bacterial community predominantly consisted of a specific cluster within the Oceanospirillus, later dubbed Deepwater Horizon Oceanospirillus. The features of its genome reveal how this bacterium grows efficiently on alkanes. As hydrocarbons are poorly soluble in water, this bacterium encodes extensive exopolysaccharide production and pili through which it can attach to the oil-water interface. Biosurfactants can be produced that emulsify the alkanes and increase the oil-water surface area. The genome contains several regions of atypical GC content, one of which carries a complete gene set for alkyne degradation. The community soon changed in mid-June to a community where most bacteria hailed from the genera Cycloclasticus, known to be obligate degraders of aromatic hydrocarbons, and Cowilia, a genus of psychrophilic marine heterotrophic generalist. Diversity increased considerably by early September and included various alpha proteobacteria, several lineages of gamma proteobacteria, flavobacteria, actinobacteria, planktomycetes, chloroflexi, and the SAR406 cluster. Petroleum hydrocarbons are a natural occurrence in all marine environments, which has allowed for various microorganisms to evolve and use these hydrocarbons as carbon and energy sources necessary for growth. Their rapid evolution is a result of their fast growth and the single copy of DNA they have, causing any mutations and gene transfers to go in effect immediately. Just like your automobile, these marine-dwelling bacteria use hydrocarbons as fuel and emit greenhouse gas carbon dioxide as a result. The microbe breaks down the ring structure of the hydrocarbons in sea oil using enzymes and oxygen in seawater. Microbes breathe in oxygen and burn the hydrocarbons from the oil in order to make sugars, fats, and proteins needed for the growth and energy production. Because of the immense amount of oil from the spill, more microbes will come to utilize the oil. When all of these microbes are in the same spot trying to consume as much oil as they can, oxygen levels will decrease because oxygen is required in order for these microbes to essentially breathe. This is why biodegradation of any chemical is mostly seen with a decrease in oxygen that can potentially have an effect on other organisms in the area. Polycyclic aromatic hydrocarbons are found in low levels of crude oils. However, they are toxic both to animals and plants. Bacteria can convert these PAHs to biomass CO2 or H2O, but first they require an insertion of O2 via dioxygenase enzymes. It is possible to continue hydrocarbon degradation at a much slower rate without the presence of oxygen. In addition to oxygen, other elements required for microbial growth includes nitrates, phosphates, and iron, all of which can potentially limit the rate of oil biodegradation, slow bacterial division, and decrease bacterial counts. Another factor that contributed to growth of the bacterial communities was the temperature of the water. In one specific example, the microbe, Cowilia, was found to be more abundant in water that was 4 degrees Celsius than water that was of a warmer temperature. This implies that the current temperature of the water plays a big role in the consumption of the oil. In an attempt to aid bacterial communities in degrading the hydrocarbons, humans added several dispersants, such as dioctyl sodium sulfoxanate, near the spill site in hopes that they would increase the available surface area of the oil, therefore increasing the rates of biodegradation. After the microbes have finished degrading the oil, there is some end product to consider such as carbon dioxide and water. In addition, Some of the oil and its components are only partly broken down, and those may be toxic to other organisms. However, the higher trophic levels are most likely to be unaffected by these byproducts of oil consumption from their microbes. In conclusion, microbes play a significant role in the degradation of many different types of hydrocarbons, specifically with oil. This process has become one of the most important tools in bioremediation and aids in tackling similar future events.